Welcome to Science with Tal. In today's video, we will cover the auditory system and understand the physiology that makes it possible for us to hear. With respect to the auditory system, there are three main topics that we will focus on. First, we will go through the anatomy of the ear to get us situated. Then, we will cover the transduction mechanisms inside the ear that convert sounds into electrical impulses. And finally, we'll end by going over how this system is organized in the central nervous system. Now, in order to understand how our ears can transform sounds into information that our brains can interpret and act upon, we first need to understand what sound is and some of its general properties. The reason why we do this is to introduce a bit of terminology that we will use throughout the rest of the video. So, to illustrate these properties, let's imagine a tuning fork near us, which will represent a sound source. If we hit this tuning fork, sound waves will propagate from this source, and this is what we will be able to hear. Now, if we pause for a second, what these sound waves truly represent can be understood if we consider the air molecules that carry the sound. As you can see, there are regions where the air molecules are more close to each other and zones where they are further apart. The zones where the molecules are more condensed and thus exert a higher pressure are called compressions, whereas zones with lower pressure are called rarefactions. An important property about these compressions and rarefactions is that they alternate, and their traveling alternance is what conducts the sound to our ears. Because of this alternance, another way to visualize sound waves is through a sinusoidal curve. Naturally, the peaks of the curve correspond to the compressions and the troughs to the rarefactions. To avoid any clutter, we will use these sine curves to understand the remaining sound properties, but keep in mind that the peaks are compressions and the troughs are rarefactions. Now, if we stamp this curve and change our tuning fork, there are a few more properties that we can discuss by hitting the tuning fork again. First, we can mention the amplitude, which is defined as the maximum displacement of vibrating particles from equilibrium. The unit associated with amplitude is the decibel and is abbreviated as dB. Graphically, it will represent the height of the peak. The amplitude of a sound will directly determine its loudness, so sounds with higher amplitude will be louder. In this case, the blue wave that we have seen in the previous example has a higher amplitude and thus will be louder to the listener. The second property that we can see from this wave is the frequency, and it is defined as the number of cycles per second. This measure is expressed in hertz, and conventionally, one cycle is the time interval between two peaks. The frequency will determine the pitch of the sound, so sounds of higher frequency will have higher pitch. In our example, you can see that the red wave has a higher pitch than the blue wave because more cycles of peak to peak happen in the same amount of time. The third property that will be relevant for us is the phase of a sound. To put it simply, the phase can be understood as the location of a given point in the cycle. As you can see, the red and blue curves have the same starting position, but if we consider a random point in time, you can see that the curves will be at a different portion in their cycle. For example here, the red curve is at its peak, while the blue curve is at its trough. This property is not super important yet, but you will see later that the brain uses the difference in phase to localize sounds. Finally, the last property to cover is not a property per se, but rather a bit of terminology I want to briefly discuss. This aspect in question is the Fourier transform, and to understand the intuition behind it, I want to switch the sound produced by the tuning fork by something we are more accustomed to in our daily lives. So, although the sinusoidal curves I just showed are good to understand the basic properties of sound, when you hear my voice or any other noise that is more than just one frequency, the wave looks more like this. As you can see, such a wave has a varying frequency and a varying amplitude across its duration. Accordingly, such a sound, in this case speech, is referred to as a complex sound. Now, with respect to the Fourier transform is a mathematical algorithm that decomposes these complex sounds to their more simple components. So, in other words, this wave of varying frequency and amplitude can be decomposed in the sum of simple curves with different properties. Inversely, the simple curves can combine to make the complex sound again. As we will see throughout the video, our auditory system is basically set up as a Fourier transform. It takes the complex noise from our environment, decomposes them into their simpler elements, and then processes it back again for us to properly hear our surroundings. With this being said, now that we understand these properties, let's discuss the anatomy of the ear. So, when it comes to the anatomy of the ear, this organ can generally be divided into three sections: the outer, the middle, and the inner ear. The outer and middle ear are mostly responsible for transmitting the sounds from our environment to the inner ear where the transduction process happens. Transduction, in other words, is the conversion of the mechanical stimuli from sound to a neural stimuli that the brain will be able to interpret. Let's start with the outer ear and work our way towards the inner ear, where we will naturally be able to cover the transduction mechanisms. So, the two main components that make up the outer ear are the auricle, which is also called the pinna, and the external acoustic meatus, which is often also called the ear canal or auditory canal. If we first consider the auricle, the structure represents the visible portion of the ear outside the head, and it comprises a few anatomical structures such as the helix, the antihelix, the concha, and the auricular lobule, to name a few. The shape of all these structures is mostly defined by the underlying cartilage that resides within the ear. Now, more importantly for us, the auricle as a whole performs two important functions with respect to the auditory system. First, it is the first structure that captures sound, and it allows to focus that sound on the ear canal. Secondly, the intricate shape of the auricle allows us to filter frequencies on the vertical axis, and thus allows us to determine whether sounds come from above or below us. We will get back to that aspect later. This sound that gets captured by the ear canal then gets transmitted to the tympanic membrane, or simply eardrum, where it will connect with the middle ear. In terms of function, there are two notable roles that the ear canal plays in this system. First, along with the auricle, the ear canal focuses and boosts the sound that travel towards the middle ear, and it does so best for sounds around 3 kHz. Secondly, the ear canal plays an important role in protecting the ear from foreign bodies or infections by producing cerumen, which is also known as earwax, via the ceruminous and sebaceous glands. Finally, two structures surrounding the outer ear are relevant to point out, namely the temporalis muscle and the temporal bone. Accordingly, problems with these structures may be the source of auditory deficits. Now, if we follow the tympanic membrane, we arrive at the middle ear, where, again, the purpose is to transmit sounds from the outer ear to the inner ear. To do so, there are 6 essential structures that work together to make this happen smoothly. First, there is the tympanic membrane that we have seen previously in the outer ear. Then, there are the 3 small bones, also called ossicles, that link the tympanic membrane to the oval window of the inner ear. This oval window is not something that we can clearly see from this view, but we'll get back to this when covering the inner ear. For the moment, we can keep in mind that this window serves the purpose of connecting the middle ear with the inner ear. Now, the 3 ossicles that I just mentioned are respectively called malleus, incus, and stapes. Additionally, the middle ear contains 2 important muscles called the stapedius and the tensor tympani. Finally, the middle ear is connected to the nasopharynx by the auditory, or sometimes called the eustachian tube. To clarify where this tube leads to, you can consider this sagittal view of the upper neck area, where you can see a bit better the opening of the auditory tube into the nasopharynx. Alright, so to make sense of all these structures and how they work together, let's cover them gradually, starting from the tympanic membrane. As we've seen in the outer ear section, the tympanic membrane receives the sound vibrations that are funneled by the outer ear, and its purpose is to transmit those vibrations to the ossicles. If we take a look at the tympanic membrane more closely, from the point of view of the ear canal, this is what it will look like. This is, by the way, the view that one would see using an otoscope, which is the typical instrument used in the clinic to observe the outer and middle ear. In this view of the tympanic membrane, there are a few landmarks that I think are useful to point out. They are not super important to know to understand the auditory system, but there are key landmarks to recognize if you study in medicine, for example. So, very briefly, the tympanic membrane is made principally out of 2 parts, the pars tensa and pars flaccida. In comparison to each other, the pars flaccida is a loose tissue, while the pars tensa is more dense and forms the annulus that surrounds the membrane. In the case of a perforated eardrum, these 2 are the tissues that will most likely be involved. In the middle of this membrane, you can see the handle of the malleus and the umbo, which is the most depressed part of this ossicle. As you can see, the malleus is directly attached to the membrane, and this is super important to faithfully transmit the sound to the inner ear. Finally, I want to mention the cone of light that typically appears in a healthy tympanic membrane under the otoscope light. If this cone of light is absent or displaced, it could suggest an underlying problem with the middle ear, for example, an accumulation of liquid due to an infection. Now, as I mentioned, the tympanic membrane transmits the sound vibrations to the ossicles, which then transmit the vibrations to the inner ear. Naturally, one could ask itself, what is the purpose of these ossicles? Why not have the membrane directly be a part of the inner ear? And what benefits do these small bones provide to the auditory system? To answer this, there are two main elements that I want to consider. First, I want to make a small aside and consider the concept of acoustic impedance. By definition, acoustic impedance is the tendency of a medium to oppose the propagation of a sound. As such, different mediums, for example solids, liquids, or gases, will propagate sound differently. The reason why this concept is important for us is because if we consider the sound that reaches us at the ear canal, this sound travels in air, a medium with low impedance, and thus makes it easy for the sound to propagate. However, as a little preview of what we will discuss shortly, the inner ear, where sound gets converted into neural signals, is an organ filled with fluid, and thus, in comparison to air, it has a much higher impedance. Accordingly, sound waves have a much harder time to propagate. Conceptually, the problem with sound going from a lower to a higher impedance is that the transmission of sound will inevitably diminish as it crosses mediums. For example, most airborne sound energy gets reflected by water. This explains why, let's say you are fully submerged in a pool, you will hardly understand the people speaking out of the water because the sound waves they produce outside get reflected by the water, and thus, not much sound energy reaches you. In the same fashion, this problem is very important to consider in the ear because this impedance mismatch between the environment and the inner ear causes a drop of around 30 decibels. To give you a rough idea, a loss of 30 decibels is equivalent to a moderate hearing loss. Thankfully, our bodies have developed compensation mechanisms to fix this mismatch. First, the ear canal, as we've seen, is a first step that allows amplification of the sound energy as it concentrates the vibration to a small resonant space. This, however, only works for frequencies around 3 kHz, but this is good since this frequency range is mostly associated with speech. The main mechanism to even things out comes from the three ossicles, and they compensate the mismatch in two main ways. First, the arrangement of ossicles concentrates the energy of the relatively large tympanic membrane on the relatively small oval window to amplify the sound that is transmitted to the inner ear. Additionally, the ossicles move in a lever-like motion that also contributes to increasing the energy. Altogether, this amplification of pressures allows the middle ear to boost pressures up to 30-fold to match the impedance between the two mediums and thus ensure the efficient transfer of sound energy. I know this topic might be a bit complicated to understand, but the essence here is that the main function of the middle ear is to boost the sound received at the tympanic membrane to avoid losing any sound energy. Back to this diagram of the middle ear, the second reason why ossicles are important in this system can be understood by considering the two muscles in the middle ear that I've mentioned, the stapedius and the tensor tympani muscle. When we consider the full path of the tensor tympani, this muscle connects to the malleus, while the stapedius, as its name implies, connects to the stapes. These two muscles, while they connect to different ossicles, perform somewhat similar functions. In both cases, these two muscles contract and allow to dampen loud sounds by diminishing the movements of the ossicles. The tensor tympani, which is innervated by the fifth cranial nerve, pulls on the malleus and thus reduces the movement of the eardrum. The stapedius, which is innervated by the seventh cranial nerve, pulls on the stapes to reduce oval window movements. In both cases, less movement of the middle ear reduces the energy that is transferred to the inner ear. This mechanism of sound dampening is also known as the acoustic reflex and is super important to protect the ear in the case of loud sounds. Two caveats with this reflex, unfortunately, is that the muscle contractions are relatively slow, so a very sudden sound, such as an explosion, will be faster to do its damage before the muscles can contract. Secondly, these muscles get tired rather fast, so sustained loud sounds will also end up being damaging to the ear. So, in summary with the ossicles, when we think about why they are important especially important because they allow to amplify the sound energy that is transmitted from the outer ear to the inner ear and thus avoid energy loss. Also, they are an ideal site to protect us from loud sounds via the acoustic reflex. Finally, to conclude this topic on the middle ear, I want to briefly discuss the auditory or eustachian tube, which we have seen connects to the nasopharynx. This tube serves two main roles. First, it allows to equalize the pressures between the middle ear and the atmosphere. This aspect is super important because for everything that we have just mentioned about the ossicles and the amplification of sound to work properly, the pressures inside the middle ear and the environment must match. Accordingly, if there is a mismatch of pressures, the middle ear works less efficiently and causes hearing loss. As such, the eustachian tube can open when needed and provide a fix for this potential problem. Otherwise, the tube remains closed. Lastly, the eustachian tube can also serve as a conduit that drains the mucus that might form in the middle ear in instances of infection. With this being said, this concludes our discussion on the middle ear. Let's now move to the inner ear by following the stapes and the oval window. This is where the sound that we have transmitted from the environment to the outer ear and then to the middle ear reaches the organ that will convert it to neural signals. Now, there's a few ways we can discuss the inner ear, either functionally or structurally. Luckily, these two kind of go hand in hand together, but if we start structurally, the inner ear can be divided into three sections: the semicircular canals, the vestibule, and the cochlea, where our sound waves will be transduced into neural signals. We can also note the round window that is part of the vestibule. So, in comparison to the two previous divisions of the ear that we have covered, the inner ear is involved in more than just the auditory system. Indeed, it is also part of the vestibular system. For that reason, I want to first describe the structures that make up the vestibular system to get an idea of what this system performs, but the details about how everything works will come in another video. To do so, let's consider what we can find inside the inner ear. As you can see, there are two main types of structures, which, more precisely, are referred to as labyrinths. The outermost labyrinth is known as the bony labyrinth and contains perilymph, while the innermost labyrinth, in light violet, is the membranous labyrinth and contains endolymph. The difference between endolymph and perilymph is not super important for us at the moment, but we will come back to the properties between the two when discussing the transduction mechanism of the cochlea. Now, to make it simple, the vestibular system is responsible for processing sensory information that relates to self-motion, head position, and spatial orientation. These sensations are then integrated in motor responses that help stabilize gaze, head position, balance, coordination, and overall posture. The organs responsible for picking up these sensations include the utricle, the saccule, and the semicircular canals. The utricle and saccule detect linear movements of the head, with the utricle sensing primarily horizontal accelerations, while the saccule senses primarily vertical accelerations. The semicircular canals are responsible for picking up rotational movements of the head. All these sensations from these organs are picked up by the vestibular nerve and are carried to the central nervous system. Again, much more needs to be said about the vestibular system, but it will be covered another time. Now, for the auditory system, what is left to mention is that it also has its own nerve, called the cochlear nerve, and as you can expect, it picks up auditory information and carries it to the central nervous system. The cochlear nerve and the vestibular nerve combine together to make the 8th cranial nerve, the vestibulo-cochlear nerve. Alright, for the most part, we have covered all the important anatomy landmarks of the outer, middle, and inner ear. As a quick recap of what we should know for the moment, the sounds that come from the environment are defined by a few key properties, namely their amplitude or loudness that is expressed in decibels, and their frequency or pitch that is expressed in hertz. These sounds reach the outer ear where they will be funneled by the ear canal to the tympanic membrane. By way of moving the tympanic membrane, the ossicle move as well inside the middle ear and allow to amplify and propagate the sound energy to the inner ear. Now, what we are ready to understand is transduction, which is simply the process of converting an external stimuli, in this instance sound, a mechanical stimuli, into a neural signal that our brains will be able to interpret. To do so, we'll first need to make a few adjustments to our diagram to make things simpler and intuitive for us. First off, since the transduction mechanism mostly involves the inner ear, we can disregard most of the ear components we have. Also, since we are not going to discuss the vestibular system, we can remove the semicircular canals and simply consider the middle ear components, vestibule, and the cochlea. Then, you can see that the cochlea is all coiled up on itself. It will help us to uncoil it and consider it as a tube. Finally, I want to shift the drawing a bit, as it will allow us to remove the vestibule and better see what's inside the cochlea. So, just to situate ourselves a bit, you will recognize the oval and round windows that are attached to the vestibule. Additionally, the pole closest to the vestibule is called the base, while the other pole is called the apex. As you go from the base to the apex, the diameter of the cochlea diminishes. Now, let's take a look at the cross-section of the cochlea to understand how it's organized. So, the cochlea is divided into three main liquid-filled compartments: the scala vestibuli, the scala media, and the scala tympani. The Reisner's membrane separates the scala vestibuli from the scala media, and the basilar membrane, as shown in red, separates the scala media from the scala tympani. This red basilar membrane is what is highlighted on our cochlea. You can see that it has a varying shape throughout the length of the cochlea, and we'll come back to this aspect later to understand how it impacts transduction. From what I mentioned, you can also notice that the scala vestibuli is above the basilar membrane, and the scala tympani is below the membrane. The two liquids merge at the apex at a region called the helicotrema. Now, for the liquid itself, both the scala vestibuli and tympani are perilymph, which is a sodium-rich and potassium-poor liquid. On the other hand, the scala media, which is also known as the cochlear duct, is filled with endolymph, a sodium-poor and potassium-rich liquid. As you will see shortly, these two different liquids will be important for the transduction process. In the cochlea, there is a third membrane to consider, namely the tectorial membrane here shown in orange. This membrane is very important as it is part of the organ of Corti, the site where the transduction of sound to a neural stimuli actually occurs. You can see on this zoomed figure that the organ is made out of a wide variety of cells which all play their role to transduce sound. Lastly, the neural stimuli are then carried out by the spiral ganglion to the cochlear nerve, where it will reach the central nervous system. Alright, now that we have established some of the important terminology in the cochlea, let's see the different steps that take place to transduce sound into a neural signal. To do so, we will actually start by taking a step back from the cross-section and simply consider our cochlea and red basilar membrane. When we'll get a good intuition on what happens at a relatively macroscopic level, we'll dive deeper into the underlying biomolecular cascade that makes it all work. So, to make our first example intuitive, let's imagine that we have a fully uniform elastic basilar membrane. From what we know up to this point, the path of sound goes like this. It enters our ear and hits the tympanic membrane, and this sets in motion the malleus, then the incus, then the stapes, which hits the oval window. Now, you may recall from our early topic that sound is made out of compressions and rarefactions. Compressions are regions of high pressure and with respect to the sine wave correspond to the peaks, whereas the rarefactions are regions of low pressure and correspond to troughs. Accordingly, we have two situations in this example. What happens to the basilar membrane when a compression hits the tympanic membrane, and what happens when a rarefaction hits the membrane. You can see between the two cases that the sounds are at different points in their cycle. One is at its peak and the other is at its trough. As such, if we consider the compression scenario first, the sound energy transferred from the tympanic membrane during a compression will drive the ossicles, particularly the stapes, forward and thus exert a pressure on the oval window. Accordingly, the pressure received by the oval window will propagate to the scala vestibuli and increase the pressure inside this cavity. This pressure will then propagate via the helicotrema to the round window, which will absorb that added pressure and as a result will bulge out. The net result of this pressure change will be the downward movement of the basilar membrane. In opposition, a rarefaction will drive the stapes outward, thus decreasing the pressure in the scala vestibuli. The compensating response will be the inward bulge of the round window and the upward movement of the basilar membrane. An additional layer that we can add to this is the concept of frequency and amplitude. Recall that frequency is defined as the amount of cycles per second, with a cycle being the interval between two peaks, and amplitude is defined as the maximal displacement of vibrating particles from equilibrium. Based on what we said, this means that the frequency of a sound will determine how many times this uniformly elastic basilar membrane goes up and down, and for amplitude, since it directly impacts how much pressure goes in and out of the cochlea, sounds with higher amplitude will generate stronger movements of the membrane. Now, keep in mind that this is an idealized example, and we'll get to shortly on how a real basilar membrane actually works. Nonetheless, there are some key takeaways from this easier example we can take. First, the basilar membrane is responsive to the compressions and rarefactions of sound, and thus to its frequency and amplitude. Secondly, the round window and helicotrema are crucial in compensating the pressure changes as sound hits the cochlea. Alright, let's now consider how our actual basilar membrane works. The first important nuance between our real membrane and the previous example is that the basilar membrane is not uniformly elastic. The reason for this is that the membrane at the base is more narrow, thick, and stiff in comparison to the membrane at the apex, which is wide, thin, and soft. The implication of this varying elasticity is that the membrane will have a varying response to different frequencies. To illustrate this, let's imagine that we have a high-frequency red sound wave that hits the tympanic membrane. As we would expect, the basilar membrane will move due to the pressure changes inside the cochlea, but it will selectively do so closer to the base because the membrane at the base responds best to high frequencies. If we send a low-frequency blue sound wave, instead of moving the membrane at the base, the membrane will maximally respond at the apex because it is best tuned to low frequencies. As you can imagine, anything in between the two extremes, such as this green wave, will create a maximal response in between the base and the apex of the membrane. So, as you can see, there is a spatial arrangement of sound frequencies in the cochlea. The base responds best to high frequencies that are up to 20 kHz, whereas as you progressively move towards the apex, this region is best suited to respond maximally to low frequencies, as low as 20 Hz. The proper terminology to refer to this arrangement is tonotopy and is often illustrated as this gradient of colors. As you will see in later sections, the central nervous system carries this tonotopic arrangement as well, so we will definitely revisit this concept. Now, an important consequence of this tonotopy is that the bounds of frequencies, so 20 Hz to 20 kHz, represents our auditory range. Accordingly, anything outside those frequencies will not be heard no matter how loud it is. For example, bats, which use echolocation to navigate and hunt in their environment, produce screams that can range from 100 to 140 decibels. Sounds of this amplitude in our auditory range are comparable to concerts, jet engines, and rifle blasts. In other words, these are all very loud sounds that can damage our ears. However, these screams that bats produce are in the ultrasonic range, so beyond the 20 kHz that our ears are capped to. As such, no matter how loud they scream, we will never hear their ultrasonic screams. Now, beyond the basilar membrane being considerably more complex than in the first example, the sounds that we hear are also more complex. By definition, what makes a sound complex is the fact that they are made out of multiple frequencies. If you recall our discussion on these complex sounds in the first section, you might remember this notion of Fourier transform. As a quick reminder, the Fourier transform is a mathematical algorithm that can decompose complex sounds into their simpler components that have a given frequency. Inversely, these simpler components can be added together to remake the complex sound. Now, the reason why I mentioned the Fourier transform is because our basilar membrane basically acts like one. Because of its tonotopic arrangement, when complex sounds hit the membrane, the component frequencies of the complex sound will preferentially cause a maximal response at different places along the length of the membrane. So, as you can see, if we consider this complex wave, which is presumably made out of 3 simple frequencies, there will be 3 different peaks on the membrane that will identify which frequencies make up the sound. As we will see later, these frequencies can then be transmitted to the brain that will compute back what the complex sound is. Alright, now I know these concepts can be a bit harder to grasp, and accordingly, let me summarize some of the main points in this section. So, very simply, the vast majority of the sounds we hear in our daily lives are complex sounds, and accordingly, they have varying frequencies. When these sounds propagate to the cochlea via the outer and middle ear, the variable shape of the basilar membrane allows to decompose the complex sounds into their single frequencies by having different maximal responses. Additionally, this tonotopy is what defines our auditory range. Lastly, I didn't go over it too much, but the higher the amplitude of a sound is, the stronger the basilar membrane will move, and thus lead to more activation around the cochlea, which will allow us to perceive the sounds as louder. Now, with all this information in mind, now that we understand that the basilar membrane will move up and down at various places depending on the incoming frequencies, the natural question to ask ourselves is how is this related to the transduction of sound into neural signals. To understand this, we need to go back to our cross-section and discuss the organ of Corti, which you can see boxed in the middle of the cross-section. If we zoom on that box, this is what the organ generally looks like. To situate ourselves, let's repoint the landmarks that we already have established. In light blue, we have the scala media filled with endolymph, and in light violet, we have the scala tympani filled with perilymph. The two cavities are separated by the basilar membrane in red. You will also recognize the tectorial membrane in orange, as well as the branch of the cochlear nerve. Now, what is new about this particular diagram are the different cell types that we can see more clearly. With respect to transduction and the auditory system more generally, the two important cell types are the outer and inner air cells. Generally speaking, there are about 3,500 inner air cells along the length of the cochlea, and these are the cells that transduce sound into neural impulses. For the outer hair cells, there are about 12,000 of those, and they are generally responsible for otoacoustic emissions and cochlear amplification. These functions may seem super mysterious right now, but we'll come back to it later. Now, both the inner and outer hair cells are innervated by the cochlear nerve, and as we've seen previously, this nerve carries the neural signals transduced by hair cells to the central nervous system. What I did not mention before, though, is that there are also efferent fibers that come from the central nervous system and modulate the sensitivity of the hair cells. We'll talk more about these mechanisms later on, especially in relation to other hair cells. Finally, this diagram also illustrates a wide variety of supporting cells, such as Spiller, Dieter, Claudius, Hansen, and Butcher cells. All these cells provide support to the organ of Corti to make sure everything works properly. Alright, now that we have most of the general terminology down, we are ready to explore how air cells transduce sounds to neural stimuli on a cellular level. The first step in this process is to understand the general structure of the hair cells and the environment in which these cells reside. Accordingly, we'll need to take a closer look at these hair cells. Since the inner air cells are the ones that transduce sound, we'll focus on them during this conversation, but most of what I'll say still applies to outer air cells. So, hair cells can generally be divided into two main compartments. The first compartment, which is on the apical side, bathes in the endolymph of the scala media and is made out of the hair-like structures called stereocilia. Hair cells have between 10 to 100 of these stereocilia, and these structures are the ones that link basilar membrane movements with ionic flux inside the cell. In other words, stereocilia mediate the first step of transduction. Something you might see in your textbooks is also the presence of a kinocilium. Hair cells generally have one, and what is interesting about it is that it is a true ciliary structure in comparison to the stereocilia. Nonetheless, the kinocilium disappears shortly after birth, so its presence is not super important for us. Now, towards the basal side of the hair cell, we can find a cell body that contains all the necessary organelles to communicate with the afferent terminals of the cochlear nerve. We'll discuss how these mechanisms operate later, but for the moment, what I want to point out about the cell body is that the basolateral surfaces are exposed to perilymph, the same perilymph that fills out the scala tympani. The fact that hair cells are both exposed to endolymph at their apical side and perilymph at their basolateral surface is super important for transduction to properly function. The main reason comes from the fact that because of the different composition between endolymph and perilymph, the two liquids also have different electrical potentials. If we consider perilymph, which again is a sodium-rich and potassium-poor liquid as the reference, endolymph is 80 mV more positive than perilymph. This difference in potential between the endolymph and perilymph is known as the endocochlear potential, and it is the key battery that keeps this entire process going. To get some intuition on why this is all important, let's consider our inner hair cell more closely. So, hair cells typically sit at a potential between -45 and -70 mV at rest. The first event of transduction, as we will see later, is the entry of potassium through the stereocilia from the endolymph. This entry of potassium depolarizes the hair cell and makes it communicate with the afferent fibers. Now, as we know, endolymph is rich in potassium and 80 mV more positive than perilymph. Accordingly, endolymph is actually 125 to 150 mV more positive than the inside of the inner air cell. As a result, The combination between the high concentration of potassium in the endolymph and the drastic difference in charge creates a very high electrochemical gradient for potassium to enter the cell. This super high gradient is very important because it allows the rapid and passive flow of potassium into the cell to generate the necessary depolarization that can then initiate the steps of communicating with the efferent. The speed of this initial depolarization is crucial for auditory transduction because, as you might recall, the sounds we can hear can be up to thousands of hertz. As such, we need the first step of transduction to be as fast as possible to faithfully track the sounds we listen to. For the passiveness, this is also a super important component because it means that the hair cell does not have to invest any energy to get depolarized. An active process would have required hair cells to be very demanding in terms of metabolic resources. Hopefully by now, you get to see a little bit why the endolymph and its composition of high potassium is so crucial. Now let's turn to the perilymph at the basolateral surface to see why it's also important. So, from what we know right now, potassium enters the hair cell and depolarizes them to eventually make them signal on the afferents. As you might expect, if potassium keeps building up in the cell, the electrochemical gradient that we have on the apical side will diminish, and we will lose our fateful transduction mechanism. This is where the basolateral surface comes through. Since the cell is exposed to an environment low in potassium and rich in sodium, it offers a pathway for inner hair cells to remove their potassium to keep the gradient high at the apical surface. Here again, the movement of potassium is passive, and for the same reasons as on the apical side, it makes the entire process more efficient and less metabolically demanding for inner hair cells. So, to recap these concepts very quickly, what ionically initiates transduction is the passive entry of potassium from the endolymph through the stereocilia into the hair cell. This potassium causes a depolarization and starts the cascade that will eventually lead to the communication with afferents. To avoid building up that potassium, hair cells are exposed to perilymph where they can passively dump their potassium and keep their activity going. As such, both the depolarization and repolarization in hair cells is mediated by the passive flow of potassium. This is considerably different from the typical process that happens in neurons. Now, if we come back to this diagram, there remains one loose end about this potassium journey to explore. Indeed, now that the potassium is in the perilymph, what happens then? It turns out that the cochlea has a pretty neat mechanism to make everything work. This mechanism in question allows to close the loop via a small chunk of cells called the stria vascularis. It is located in this region, and it has the principal role in pumping potassium ions from the scala tympani into the scala media, thus producing endolymph. For your information, the stria vascularis is made out of 3 cellular layers, the marginal, intermediate, and basal cells, and they work together to pump potassium in the scala media. What is super important for us to keep in mind here is that the stria vascularis maintains the endocochlear potential by producing endolymph, and this is crucial for transduction to properly function. The last detail that I want to mention on this diagram is the spiral ligament that can be found right beside the stria vascularis. This ligament is essentially a zone of connective tissue, mostly fibrocytes, that contain capillaries and thus provide mechanical and metabolic support to the stria vascularis and basilar membrane. At this point, I know that the concept of transduction might still be elusive, but hopefully with the next few examples and explanations, all the loose ends will be tied. So, now that we have an idea of what hair cells are and the general structure of the organ of Corti, it's time to match that knowledge with what we have covered previously with the basilar membrane. As a quick recap, recall that sounds that hit our ears are made out of alternating compressions and rarefactions. Any sound can be described by its amplitude or loudness, as well by its frequency or pitch. With respect to the basilar membrane, when the compression phase of a sound hit the tympanic membrane, we have seen that it made the basilar membrane move down, and inversely, a rarefaction made the basilar membrane move up. Now, what we need to discuss is what happens with the hair cells when the basilar membrane moves up and down. Accordingly, we have 3 situations to consider: what happens at rest, during a compression, and during a rarefaction. On our hair cells, the region that will be important for this topic is this close-up diagram of the stereocilia. On this diagram, we can see the mechanoelectrical transduction or MET channels that are attached to the adjacent stereocilia via a structure called the tip link. In short, Tip links are structures that are believed to modulate the opening of med channels, and thus the initial transduction process. At rest, when there is no sound and no basilar membrane movement, 10 to 50% of the med channels are open and let mainly potassium but also some calcium inside the cell. This ionic flow contributes to producing the resting membrane potential that sits between -45 and -70 millivolts. Now, if we consider a compression, in which the basilar membrane moves down, this motion will create a shear force between the hair cells, the tectorial membrane, and the scalum media. This shear force will cause the movement of stereocilia towards the smaller stereocilia, and thus the closure of med channels via the tip links. As a result, calcium and potassium are prevented from entering, and the hair cell gets hyperpolarized. On the other hand, a rarefaction moves the basilar membrane up and creates a shear force towards the tallest stereocilia. As you can imagine, this opposite movement pulls the tip links and opens the med channels. With a bigger opening, more calcium and more potassium can enter the cell to depolarize it. So, as you can see, sounds, which again are made out of alternating compressions and rarefactions, will alternatively depolarize and hyperpolarize the cell. This is how the cochlea will be able to track frequency, but we'll come back to that element later. Alright, now that we understand this first important step of transduction, let's add some details and see what happens in the subsequent steps. So, as we know now, basilar membrane movements create a shear force on hair cells that either depolarize or hyperpolarize them. For outer hair cells, since they are attached to the tectorial membrane, the shear force is created by the movement of the hair cells on this membrane. For inner hair cells, they are not linked to the tectorial membrane. It is rather the endolymph that produces a shear force on the stereocilia. When this shear force pulls on the tip links and opens the med channels, calcium and potassium can flow in to depolarize. On the other hand, closure of the med channels hyperpolarizes the hair cells. For your information, the tip links that are believed to mediate the opening and closure of the channels are made out of cadherin 23 and protocadherin 15. The reason why these protein names might be of interest to mention is because a mutation in them can cause deafness as the hair cells become unresponsive to sound. Anyhow, if we consider the case of a depolarization, the entry of calcium and potassium elevates the membrane potential of the hair cell and leads to the opening of potassium and voltage-gated calcium channels. The potassium channels are either gated by voltage or depend on calcium, but either way, their opening allows for potassium to leave the cell into the perilymph. Recall that this step is very important to make sure hair cells have a continuous supply of potassium in the endolymph. The second type of channel that opens are the voltage-gated calcium channels, and these let even more calcium enter the cell. Paired with the calcium that enters from the med channels, all this calcium is super important because it mediates the release of neurotransmitters from the hair cell. To do so, calcium binds to a protein called otoferlin and allows the exocytosis of the neurotransmitters. Here again, otoferlin is a protein that is susceptible to mutations and thus potential deficits in hearing. Now, the neurotransmitter that hair cells release is mainly glutamate, and it is packaged in many vesicles at the basal end of the cell. Terminology-wise, you might see in your textbooks the description of a synapse between hair cells and the afferents be called a ribbon synapse. Simply put, ribbon synapses are electron-dense synapses that are ideal for rapid release and sustained signal transmission. This is obviously super important for our transduction process here, as we want our signal to be as faithful as possible to the sound we transduce. Once glutamate is released, it can bind to the NMDA and AMPA receptors on the afferent and depolarize the neuron to generate an action potential. Recall that hair cells rest at a membrane potential between -45 and -70 millivolts, and their med channels are always a little bit open. As such, even at rest when there is no sound and no basilar membrane movement, there will be enough calcium that enters to cause neurotransmitter release. This is important to keep in mind because it means that the depolarization and hyperpolarization that are caused by the movement of the basilar membrane simply modulate how much neurotransmitters are released. We will later see the implications of this detail. With this in mind, hopefully the key steps of transduction are somewhat intuitive. If not, do not hesitate to ask away in the comments. Alright, looking back on our main diagrams of the ear, let's take a minute to briefly recap what we went over. Sound that is defined by a given frequency and amplitude arrives at the tympanic membrane and gets funneled to the inner ear by the orchestrated ossicle movements. In the inner ear, which for our purposes is better visualized as uncoiled, there are two key structures that we discussed at length. First, there is the basilar membrane that moves up and down in response to sound and is divided into a tonotopic arrangement as shown by the gradient from 20 kHz to 20 Hz. The basilar membrane movements are particularly important for the second structure, which is the organ of Corti, as it mediates the transduction of sound into neural signals. This transduction is performed by the inner hair cells that then send their signal to neural afferents. Although we generally have a good idea of what happens at this level of the auditory pathway, there are still two topics that I want to cover before we start learning about how the auditory system operates in the brain. The first topic that I want to discuss is the role of the outer ear cells, which for the moment have remained rather mysterious. Then, I want to conclude by discussing how the neural innervation is organized throughout the cochlea. This will bridge nicely into our next section. So, if we go back to this diagram of the organ of Corti, we can find outer hair cells arranged in these three rows, which makes these cells between 3 to 4 times more numerous than inner hair cells. From what we mentioned previously, outer hair cells mediate otoacoustic emissions and cochlear amplification, but what does that even mean? To understand this, it's important to know that a considerable portion of the sound energy that the cochlea receives is lost in trying to move the fluids instead of exciting the hair cells. Paired with the fact that the precision of the ear is way too sharp to function purely passively, several lines of evidence indicated that there must be a mechanism in the cochlea that amplifies sound energy. This is where outer hair cells come into play. If we take a closer look at outer hair cells, you will see that they are very similar to inner hair cells. However, they do have some key differences. First, afferent fibers only make minimal contact with outer hair cells. Instead, these cells are predominantly innervated by efferent fibers that come from the superior olivary complex in the brainstem. We'll come back to this nucleus later, just keep in mind that the outer hair cells are mostly innervated by efferent fibers. Secondly, the lateral membrane of the outer hair cells is densely packed with a protein called prestin that is not present in inner hair cells. This protein in question is believed to be the key player that mediates electromotility, or in other words, the ability of the cells to change their cell length in response to varying electrical charges. Without going too much in depth into the mechanisms of prestin, as it goes outside the scope of this video, prestin is a voltage-sensitive protein that allows the lengthening or shortening of the cell body. For example, a depolarization of the cell membrane shortens prestin molecules and thus shortens the cell. On the other hand, a hyperpolarization lengthens the membrane and as a result, the cell. Given that everything is connected together in the organ of Corti, the oscillatory motion from outer hair cells is believed to provide mechanical energy to boost basilar membrane motion and accordingly sound energy. With what we've seen previously, it should be intuitive to us now that more movement of the basilar membrane will mean more sound detection, more inner cell activity, and sensitivity. Now, there are a few ways for outer hair cells to be depolarized and hyperpolarized. Of note, whenever outer hair cells get deflected such that potassium enters the cell, this contributes to depolarizing the cell. For the hyperpolarization, the efferent branches that contact outer hair cells can release acetylcholine and CGRP. These two transmitters have a hyperpolarizing effect and thus lengthen the cell. Although outer hair cells seem to play a prominent role in cochlear amplification, OHC are unlikely to be the only mechanism at play, and much more remains to be discovered about this process. The scientific literature regarding this topic is a bit nebulous at the moment, but hopefully in a few years we'll get more clarity on this entire process. Finally, besides the cochlear amplification aspect of outer hair cells, these cells are also believed to play a role in otoacoustic emissions. As the name implies, otoacoustic emissions are sounds that are produced from the cochlea, and while they can be evoked or spontaneous, outer hair cells are believed to play a role in their production. These otoacoustic emissions are particularly important in the clinic, where they are the basis for a simple and non-invasive test for cochlear hearing loss in newborn babies. Hopefully this provides some intuition on what outer hair cells might be doing in the cochlea. Alright, now that we have a good idea what role outer hair cells seem to perform, let's finish our discussion on the inner ear by tying everything up and discussing the neural innervation of the cochlea and what message is actually being sent by the hair cells. So, from our cochlea, a good way to see how the entire system is organized is by taking this cross-section. As you can see, the cochlea coils around the central axis to give its characteristic snail shape. This central axis, which is also known as the modulus, is a spongy bone that holds the neurons that innervate the cochlea. The cell bodies of the neurons are located in a particular structure called the spiral or cochlear ganglion. Accordingly, the neurons in these ganglions are called spiral ganglion neurons and converge all together to form the cochlear nerve or cranial nerve 8. This nerve is what again will send auditory information to the central nervous system. To do so, it passes through a structure called the internal acoustic meatus, which is simply a conduit that offers the passage for cranial nerve 8 and other nerves that are not super relevant to the auditory system. Now, if we consider our organ of Corti, it turns out that there are two types of spiral ganglion neurons. 90 to 95% of the afferents that innervate the cochlea are type 1 and contact inner hair cells, whereas the remaining 5 to 10% that make up the type 2 spiral ganglion neurons contact outer hair cells. For type 1 SGNs, the general rule is that one axon of an SGN contacts one inner hair cell, but inner hair cells can activate around 10 axons. Each type 1 SGN responds best to a characteristic frequency and follows the tonotopic order that is set in the cochlea. You will see shortly that these two details will be important to consider. For other hair cells, although they send some information that is picked up by these type 2 SGNs, we unfortunately do not know much about what this information codes for and whether or not it is used for auditory processing. With respect to the efferent innervation, mature inner hair cells do not receive any efferent input directly. For other hair cells, however, we've seen that they are the primary cell targeted by these efferents, and it plays a role in this entire cochlear amplification mechanism. Now, with this in mind, we know what the innervation of the cochlea looks like and what type of connections are made on our hair cells, but we have yet to discuss what exactly is the signal that is sent from hair cells. Given the rather mysterious nature of other hair cells, let's focus on what is carried by type 1 SGNs. So recall that in the entire process of sound transduction, we essentially have three phases. We have a rest phase, where the basilar membrane is not displaced. Since the med channels are always open at rest, glutamate gets released and excites the afferents. When sound energy hits the cochlea, it can either displace the basilar membrane up or down, depending if the sound is in a compression or a rarefaction phase. If it's in a compression phase, we've seen that the basilar membrane moves down, the stereocilia close, and the inner hair cells gets hyperpolarized as a result. This hyperpolarization then causes the decrease in glutamate release and thus activity in the afferent. On the other hand, a rarefaction causes the opposite as the basilar membrane goes up, the med channels open up, and the cell gets depolarized, thus increasing the amount of glutamate released. Because of this switch between rest, depolarization, and hyperpolarization, inner hair cells are said to have a biphasic response. Now, if we imagine the simplest sound wave and we think about the patterns of activity in type 1 SGNs, then logically, the activity of the afferent should follow the phases of the wave. When the sound is in a rarefaction phase, the activity of the afferent is highest, but as the sound progresses to a compression, the activity decreases. As such, the activity of the afferent can follow and encode the frequency of the sound throughout its firing activity. However, this system, which is called phase locking, only operates up to 3 kHz. After that threshold, the information about frequency gets lost because the frequency is too high and the cells cannot keep up. We know, however, that we can hear sounds up to 20 kHz, so there must be something else compensating for this. The other system compensating for sounds above 3 kHz is called label line coding and works on the principle that every inner cell is most sensitive to a stimulation at a specific or characteristic frequency. This is very similar to the basilar membrane that had a gradient of frequency sensitivity throughout its length that was created because of its varying shape. Since a given type 1 SGN connects to one inner hair cell, it makes it that the basilar membrane, the inner hair cell, and the afferents all have a characteristic frequency. In other words, the tonotopy that is illustrated on the basilar membrane follows at all levels of the organ of Corti. This is particularly interesting from a coding perspective because if the brain knows where the SGN activity comes from, then it can reliably know what frequency has stimulated it. One way to think about it is to imagine each hair cell as a piano note and the afferents as the strings of the piano. If you know to which piano note a given string connects, then you can predict what frequency is played when the string moves. Now, from an experimental perspective, if we consider the stimulus magnitude required to stimulate type 1 SGNs to a standard level against the frequency of sound that is played, you will see the following result. Each afferent has a given characteristic frequency that requires the least amount of intensity to fire the afferent to the standard level. For example, if we look at the green curve, we see that for this given type 1 SGN, you can play a very faint 2 kHz sound and it will display the same level of activity that if you played a very loud 5 kHz sound. Thus, this afferent has a characteristic frequency of 2 kHz. The same logic goes for the other curves that we see on the graph. So, to quickly resume, we have two ways of encoding sound frequency. We have the phase locking system that only works for up to 3 kHz and can accurately code the frequency of the sound throughout the activity patterns of the afferent fiber. In complement to this system, we also have label line coding, which works for sounds up to 20 kHz and basically encodes the frequency of sound via the location of the afferent. So, if an afferent innervates a hair cell that best responds to 10 kHz, although the signal won't faithfully track the 10 kHz, its location will communicate that it responds best to those 10 kHz. Hopefully, this makes sense. Let me know in the comments if there is any confusion. Finally, I want to mention that frequency is not the only important property of sound that we care about. There is also the amplitude of the sound, or in other words, how loud it is. Thankfully, the amplitude is a bit more straightforward to interpret. To put it simply, the louder the sound is, the more of the cochlea it recruits. We've seen with the basilar membrane that louder sounds will make other parts of the membrane move as well, and this is simply because it provides more energy to the cochlea. In the same fashion, loud sounds will recruit more hair cells and thus more afferents. You can even infer this relationship from the experimental graph. If we go back to our example of the green curve, you can see that it can still respond to other frequencies, but the sound has to be much louder. So if the brain receives some activity from the 2 kHz afferent, but a 5 kHz sound is playing, this in part will be able to tell that the sound playing is loud. Alright, looking back at our diagrams of the ear one last time, let's quickly resume what we have covered before we head to the central nervous system. So, very simply, sound is made of a given frequency and a given amplitude, and it is funneled and boosted in energy by the outer and middle ear to reach the inner ear. The inner ear, which we can uncoil to see better, contains the tonotopically organized basilar membrane and organ of Corti. When sound energy reaches the inner ear, the energy creates motion that inner hair cells transduce into neural signals. The transduction is transmitted to type 1 sensory ganglion neurons that leave the cochlea to reach the central nervous system. The information these neurons carry encodes the frequency of the sound directly if the sound is less than 3 kHz and indirectly based on the location of the innervation. These two systems are respectively referred to as phase locking and label line coding. In addition to this, the population of afferents also carries information about the amplitude of the sound depending on the recruitment and intensity of the signal. Now, let's see how the brain is organized to process all that information. So, to start our discussion on the auditory system in the central nervous system, I want to begin with an overview of the entire pathway, which will help us ground some key aspects of the auditory pathway. To do so, let's consider this coronal cut of the brain. The first important aspect about the auditory system is that, as with any other sense, the pathway to reach the cortex is partitioned across many levels. In the case of the auditory system, there are 5 main levels where information gets transferred and integrated before it reaches the cortex. Given their position under the cortex, these areas that are shown by the circles on the brain are all referred to as subcortical areas. If we take a look at some cross-sections of these subcortical areas, here is what the pathway looks like from one ear. As you can see, the auditory pathway makes bilateral connections early on and runs in parallel via multiple connections. This is important, and what it implies is that moving forward, we can't just simply understand the pathway on one side and mirror it, Instead, we have to consider how both ears integrate the information together. This bilateral integration, along with the parallel pathways that are created in the subcortical levels, are central to process the many aspects of sound. Now, to get more familiar with the pathway, let's see the key structures for each of the subcortical levels. It goes without saying that we will revisit each structure on their own later. So, coming off the 8th cranial nerve, which carries auditory information, the first point of entry for the auditory information is the cochlear nuclei. The two cochlear nuclei are located in the rostral medulla, and it is from there that the many parallel pathways originate. A very important aspect about this nucleus and the auditory system as a whole is that tonotopy is maintained throughout the pathway. In other words, the neurons in the cochlear nuclei are arranged in a gradient of frequency in a similar fashion to the basilar membrane and the organ of Corti. This tonotopic arrangement is not only present in the cochlear nuclei, but also in later structures and even in the cortex. As we progress to the next cross-section, we reach the superior olivary complex located in the mid-pons. This very interesting center in the brainstem is responsible for integrating bilateral information coming from the cochlear nuclei to localize sounds. As you can see, both centers receive connections from a particular ear, and we'll later see the implications of that. Moving up, the output of this complex and all the other fibers reach the lateral lemniscus and associated nuclei. The nucleus of the lateral lemniscus is believed to be associated with the processing of sound onset, its duration, and more. Unfortunately, the role of this nucleus is not fully understood, and as a result, we won't be spending too much time covering it. Nonetheless, all the parallel projections coming through the lateral lemniscus and originating at the subcortical levels converge to the inferior colliculus in the caudal midbrain. This area, just like the cochlear nuclei, is known to have preserved tonotopy and is important for sound localization in space. It is also responsive to complex temporal patterns and more. After the inferior colliculus, the next level is the thalamus and then the cortex. As we get to those areas later, we'll talk more in detail about their roles and structures. Nonetheless, to get a general idea, the thalamus is often described as a relay station that sends all the subcortical signals to the cortex, and the cortex is part of the pathway that will do the heavy processing to generate our conscious perception of sound. Now, the fourth important aspect of the auditory system is that although this is a sensory pathway and we expect information to go up in the brain, it turns out that the auditory system also has some important descending projections. These descending projections originating from the cortex go back to innervate pretty much all levels of the pathway, namely the thalamus, the inferior colliculus, the superior olivary complex, and even the cochlear nuclei. As we will see later, these descending tracks are very important to add another level of sensitivity and regulation in auditory processing. The fifth and sixth important points, which kind of go hand in hand together, are that sound is complex and holds many features that must be represented in the brain, as well as the fact that the auditory system integrates with the rest of the brain. Although these 2 points seem pretty obvious, they have some very important considerations. For example, if we take the fifth point first, sound in our daily lives can be speech, it can be noise from the environment, but it can also be music and much more. Accordingly, it is safe to assume that our brains need the processing capacities to distinguish all these types of sound, which is obviously great for our lives, but it becomes complicated to grasp when discussing how it all happens in the brain. In addition to that, our auditory perception is usually much more than a simple passive process. A great example of this is speech. When we talk, the voice that we hear is usually used to give us ongoing feedback. If our voice is too high-pitched, too loud, or too fast, we might want to decrease these characteristics to fit the social environment we're in. From the perspective of the brain, you can imagine that there must be some feedback connections that allow a continuous monitoring between the motor and the auditory aspects of speech. This notion is even more evident when someone sings, where rhythm, volume, pitch, and so on are crucial for the performance of the singer. This example is also exactly what I mean by the fact that the auditory system integrates into the brain, as auditory processing is usually subject to our attention, our memories, our environment, our movement, and so on. Other examples of this integration can be whenever you listen to a song you like or don't like, where although you are using your auditory system to process the music in both instances, there is an added layer of emotion that processes what you're listening. As such, here again, the auditory aspect is connected with other regions of the brain. Examples of that nature are pretty much endless, and the point I want to make here with them is that this section about the auditory system and the central nervous system should not be seen as a finality, but rather an opening to other systems. It also allows me to say that there is still so much that we have to learn about the processing of sound in the cortex, and unfortunately, this video will not even scratch the surface. With all this established, we should have all the proper background to dive into the pathway, starting with the first two levels, the cochlear nuclei and the superior olivary complex. So if we start at our cochlea, the afferents coming out of it are 90 to 95% the type 1 sensory ganglion neurons that innervated inner hair cells, and 5 to 10% the type 2 that innervated outer hair cells. The first target to be reached by the afferents are the cochlear nuclei, which are divided into the dorsal cochlear nucleus and the ventral cochlear nucleus. The ventral cochlear nucleus can be further divided into a posterior and anterior part, but we will not consider in great detail these two further divisions. For both the dorsal and ventral division, the nuclei preserve tonotopy, which each responding best to high frequencies towards the dorsal side and responding best to low frequencies towards the ventral side. In this figure, the ventral side is towards the bottom of the screen and the dorsal towards the top. Now, one reason why it's important to distinguish between the dorsal versus the ventral division of the cochlear nucleus is because each nucleus is made of a different population of cells that make different parallel pathways. Accordingly, the dorsal nucleus mediates different functions from the ventral nucleus. If we start with the dorsal division, it is mostly made out of fusiform cells, which send their projections to the contralateral side via the dorsal acoustic stria, or simply the DAS. In terms of the processing in that region, the dorsal cochlear nucleus is believed to integrate somatosensory, vestibular, and auditory information together, as well as processing vertical localization of sound. We'll get back to that aspect in just a minute. In the ventral division, there are three types of cells, and to better understand their roles and pathways, it's best to first discuss the superior olivary complex, which we can find in the upper cross-section. So, as we said, this region integrates bilateral information, and it is important for sound localization. More precisely, it is important for localizing sounds on the horizontal plane, and it does so by two mechanisms: interaural time difference, or ITD, and interaural intensity difference, or IID. ITD is processed by the medial division of the superior olivary complex and works for sounds under 3 kHz. IID is processed by the lateral division and the medial nucleus of the trapezoid body, or MNTB, and works for sounds above 2 kHz. We'll obviously discuss in more detail how these mechanisms operate, but the important part for the moment is to understand that ITD and IID are made possible from the connections of the ventral cochlear nucleus. More exactly, it is the bushy cells in the VCN that form the connections to make both the ITD and IID by innervating both ipsilateral and contralateral superior olivary complexes. The region at which the fibers decussate to reach the contralateral field is called the trapezoid body, or sometimes the ventral acoustic stria. The projections from bushy cells will be super important for us when discussing IID and ITD. Now, in addition to bushy cells, the VCN is also made of stellate cells, which terminate widely around the region, including the dorsal nucleus. Finally, the VCN is made out of octopus cells that terminate on the contralateral nucleus of the lateral lemniscus. The projections from the octopus cells make the intermediate acoustic stria, or IAS, and since they don't integrate bilateral information, the IAS is also referred to as a monoaural pathway. Alright, now that we have a good picture of how everything is organized in this region, let's consider the pathways of sound localization. From what we said, sound can be localized on the horizontal and vertical planes, and this information is processed in different regions. The exact circuitry that processes vertical localization is not well understood, but what is known is that depending on where the sound originates in space, the sound wave that will be registered at the ear will be modulated by the shape of our bodies. In other words, the irregular shape of our outer ear, along with the shape of our head and shoulders, create distortions in the sound that give spatial cues on where the sound might originate from. In this example, if we imagine that the same sound is being played at every angle, the varying shape of the signal is presumably used by the dorsal cochlear nucleus to, at the very least, initiate the localization along the vertical plane. Again, the actual circuitry underlying this processing is not fully understood, but when it comes to horizontal localization, we know a bit better how it seems to be orchestrated. So, if we start with ITD first, as we said, this mechanism works best for sounds under 3 kHz, and that is because this mechanism uses phase-locked information. If you recall from our discussion on inner hair cells and sensory afferents, phase locking referred to the ability of afferents to follow the compressions and rarefactions of the wave in their signal. So when the phase of a sound was a rarefaction, the activity of the fiber was highest and lowest during a compression. As such, the frequency of the sound was encoded in the activity of the afferent fiber, but after 3 kHz, the frequency becomes too high to follow. Now, if we consider a small diagram of the head and a sound source, what ITD truly represents is the difference in time between sound coming from the ear nearest to the sound source, so in this instance the left ear, versus the ear furthest from the sound source, here the right ear. As you can imagine, if the brain is able to compare and decipher that sound arrives at the left ear first in this instance, then it will be able to know that the sound source is on the left of the head. The main structure underlying this ability, or at the very least initiating this process, is the medial portion of the superior olivary complex, or simply the MSO. The processing that the MSO performs is also known as the delay lines model, and its pathway goes as follows. If we consider the right ear to be blue and the left ear to be red, bushy cells from the right will connect to the ipsilateral and contralateral MSOs, and this connection will be mirrored from the left ear. The output of the MSO will then be transmitted to the rest of the auditory pathway. If we zoom on the MSO of the left side, you can see that the ipsilateral bushy cells connect to the lateral side of the MSO, and the contralateral fibers that cross at the trapezoid body contact the medial side of the MSO. Inside the MSO, there is a population of bipolar neurons that receive the connection from the lateral and contralateral fibers. Now, as you can see from the zoomed figure of the MSO, the ipsilateral and contralateral fibers are quite different. Indeed, ipsilateral fibers are all the same length, whereas the contralateral fibers have varying length. This distinction is very important because the bipolar cells inside the MSO act as coincidence detectors, and to best understand this, let's go back to our sound source and follow the trajectory of the signal step by step. So, as the sound plays, since the left ear is nearest to the source, it will hear the sound first, and accordingly, its ipsilateral fibers will receive the signal first. As such, there will be activity on the ipsilateral side, and since we are assuming the sound is less than 3 kHz, the information received at the MSO is phase-locked, or in other words, follows the frequency of the sound. Shortly after the left ear receives the signal, the right ear receives the signal as well, and will send the signal down its contralateral branch. To see what happens next, let's focus on the zoomed MSO. So, as we said, ipsilateral and contralateral fibers have varying length. The ipsilateral fibers are of equal length, and thus, when the signal comes, it propagates at the same time to the bipolar neurons. On the other hand, when the signal arrives in the contralateral branch, it progressively passes through the bipolar neurons to excite them. This sequential activation is exactly what is meant by the delay lines. Given that the signals are phase-locked, the activation of the ipsilateral branch can miss the activation of the contralateral branch, but as the ipsilateral branch keeps getting activated and the signal keeps propagating in the contralateral branch, there will be an instance where the bipolar neuron in the MSO receives the excitation from both the contralateral and ipsilateral fiber. This simultaneous excitation of the bipolar neuron is also known as a coincidence detection, and this is what is believed to indicate the position of sound on the horizontal plane. If the sound source changes location, then the time interval between the left and right ears will change, and that will activate a different bipolar neuron in the MSO, and thus a different position will be detected. Obviously, this is just a crude depiction of this pathway, as you have to imagine the system operating on a millisecond scale and on both sides of the brainstem. Moreover, there are additional inhibitory connections in this pathway that sharpen the signal, and thus the delay lines model is much more complex than represented currently. Here, the main point is to really illustrate the intuition behind the potential mechanism that underlies ITD. With this being said, now that we have an intuition for IITD, let's see how IID is computed. This system works for sounds with frequencies above 2 kHz and depends mostly on the shape of the head. If we consider again our sound source, at frequencies higher than 2 kHz, our head starts to act as an obstacle to sound and makes a sound shadow to the ear furthest from the sound source. As such, this creates a difference in sound intensity. Accordingly, in this example, because of the sound shadow, the left ear receives the sound louder than the right ear. In a similar vein to ITD, if the brain can compare the sound of both ears and determine that the sound is louder on the left, then it can infer that the sound source is likely on the left of the head. The region of the superior olivary complex underlying this comparison is the lateral division of the complex and the medial nucleus of the trapezoid body, or simply MNTB. To best understand the IID pathway, it is best to consider both complexes. If we first consider the right ear, bushy cells provide the inputs to the pathway, with ipsilateral pathways innervating the LSO and the contralateral pathways innervating the respective contralateral MNTB. This exact innervation scheme is mirrored on the other side. In the MNTB, the bushy cells connect to an inhibitory relay that projects to the ipsilateral LSO. So, as you can see, for each side, the LSO integrates the ipsilateral excitation and the indirect contralateral inhibition from the opposite ear. Now, let's see how it all plays out with our sound source that is on the left of the head. So, as in the previous example, sound reaches the left ear first, and because there is no sound shadow, sound will be loudest on the left. In our diagram, it means that the ipsilateral and contralateral projections from the left ear will be the highest. Then, when the sound reaches the right ear, the sound will be softer, and accordingly, the activity in the projections coming from the right side will be smaller than on the left. With respect to the circuit, the activity of the lateral superior olive will be at its highest on the left side because it receives a strong ipsilateral excitation and a small contralateral inhibition. However, the dynamic is flipped on the right side where inhibition is highest, thus shutting down the projections from the right LSO. So, to put it more broadly, the louder sound excites the ipsilateral ear and inhibits the contralateral ear, thus indicating where the sound localizes. As you can imagine, this signal must also be graded to account for the sound being possibly more in front of the head or behind the head. This gradation is encoded by how much the ear is impacted by the sound shadow, and thus the firing activity of the bushy cell projections and eventually LSO neurons. Alright, with ITD and IID covered, hopefully the two pathways are somewhat intuitive. Do not hesitate to ask any questions in the comments if anything was unclear. Now, the final topic I want to mention with respect to these two cross-sections is that the superior olivary complex provides efferent feedback connections to the cochlea to regulate its sensitivity. These feedback connections primarily originate from two distinct populations, the medial and lateral olivocochlear neurons, or MOC and LOC respectively. MOC neurons innervate bilateral outer hair cells, and this connection primarily serves to modulate cochlear amplification that the outer hair cells provide. Recall that outer hair cells had a protein called prestin embedded in their lateral surface, and it allowed the cell to change size upon changes in cell charge. This process was also known as electromotility. In any case, we mentioned that when outer hair cells get hyperpolarized, their cell body lengthens, and that causes the sensitivity of the cochlea to decrease. This hyperpolarization is directly induced by the efferent input of MOC neurons that release mostly acetylcholine on outer hair cells. Decreasing the sensitivity of the cochlea can be particularly important to protect the inner ear under the stress of loud sounds. When it comes to LOC neurons, their projections innervate the ipsilateral afferents of inner hair cells. This modulation, which is performed by acetylcholine and dopamine, is a bit less understood than MOC neurons, but it seems that the two neurotransmitters provide some excitation to balance the excitability of the afferents. Unfortunately, much remains to be understood about the cochlear efferent system and how it shapes the auditory signal. Nonetheless, if we go back to this overview of the central nervous system, we should have a pretty good idea of what the superior olivary complex and the cochlear nuclei do. Let's now turn our attention to the nucleus of the lateral lemniscus and the inferior colliculus to understand what these levels do. For this, I want to consider this dorsal or posterior view of the brainstem to briefly recap our auditory pathway and get an idea of where each structure is situated. This view will look at the brainstem from this direction. So, if we start at the cochlear nuclei, we have seen that this region projects bilaterally to the superior olivary complex. The information computed in the SOC, along with the pathways from the cochlear nuclei that do not connect to the SOC, then all project to the inferior colliculus in a pathway called the lateral lemniscus. Throughout this lateral lemniscus, at the junction between the pons and the midbrain, we can find the nuclei of the lateral lemniscus, which has two main divisions, a ventral and a dorsal one. The ventral division receives predominantly contralateral monoaural input that comes from octopus cells, while the dorsal division receives bilateral input. Although we don't fully understand the roles of these nuclei, both divisions then project to the inferior colliculus in the form of inhibitory signals. As you can see, the inferior colliculus is a key region in this entire pathway because it is essentially a convergence point for all the ascending subcortical tracts that we have mentioned. It is also the target of descending feedback from the auditory cortex. Because of this information traffic, you can imagine that the inferior colliculus is super important. With respect to its function, we can infer some of the things it seems to mediate from its structure and projections. As an important relay of auditory information, certain parts of the inferior colliculus are tonotopically organized and participate in sound localization. In other parts, the inferior colliculus is responsive to complex temporal patterns and seems to filter some auditory information. Considering the downstream modulation it receives from the auditory cortex, it is not too far-fetched to imagine this region being involved in some type of filtering activity. Projections-wise, the inferior colliculus is known to project to the thalamus to reach the auditory cortex. It is also known to reach the superior colliculus in the rostral midbrain, where the integration of visual, auditory, and somatosensory input is integrated to control head and eye movement in response to visual and auditory cues in space. Finally, it is also known to project to other regions in the brainstem, including some that mediate motor and autonomic tasks. Hopefully, this segment on the inferior colliculus gives some intuition on what it does, but by no means it represents an exhaustive list of what it performs. The circuits in the midbrain can get pretty intricate. Nonetheless, going back to this diagram, we are now ready to discuss the two final levels of our pathway, the thalamus and the auditory cortex. So, from our coronal cut of the brain, if we start at the thalamus first, the more precise region of the thalamus that relays auditory information to the cortex is called the medial geniculate body, or nucleus, and represents this small bit of the thalamus. To put it simply, the medial geniculate body connects reciprocally with the auditory cortex and can be further divided into three divisions that each have their own patterns of inputs and outputs. The first division that we can consider is the ventral division, which receives predominantly input from the inferior colliculus and is thus tonotopically arranged. This area of the thalamus projects to the core, which is another way of saying the primary auditory cortex. To find the primary auditory cortex, we can consider this lateral view from which we can obtain the level of a coronal cross-section. So, the primary auditory cortex is also known as Brodmann areas 41 and 42 and lies on Heschel's gyrus. All these regions are located in the temporal lobe, medial to the sylvian fissure. So, to locate all these regions, let's start with the temporal lobe, which is highlighted here in blue. This lobe is separated from the frontal and parietal lobes by the sylvian fissure, here highlighted in black, and on our cross-section, you can see that the sylvian fissure makes this deep cut in the brain, and this is where we will find our primary auditory cortex. So, as I said, the primary auditory cortex lies on Heschel's gyrus, which is also known as Brodmann area 41 and 42. If you are unfamiliar with Brodmann areas, the areas represent a nomenclature system that divides brain areas based on cytoarchitecture, or in other words, the layering of neurons in the cortex. Accordingly, each different Brodmann area has a different pattern of neuron layering. Now, if we go back to our thalamus, the second division we can introduce is the dorsal division of the medial geniculate body, which contains more broadly tuned neurons that project to belt areas. Following the logic of the primary auditory cortex being the core area, belt areas represent the secondary auditory cortex. This secondary cortex, which surrounds the primary auditory cortex in the temporal lobe, also contains parabelt areas that surround belt areas. With respect to the Brodmann areas, the belt and parabelt areas include Brodmann area 22, which we can find mostly on the lateral surface of the temporal lobe. Finally, The third division of the thalamus, the medial division of the medial geniculate body, projects widely to the auditory areas and non-auditory areas such as the amygdala, the basal ganglia, and more. Now, when it comes to the function of the cortices we just introduced, if we start with the primary auditory cortex, it is known to be tonotopically arranged with patches of neurons that respond to both ears or only one ear. The tonotopy is arranged such that neurons towards the rostral side, so towards the nose, respond best to low frequencies, and the caudal side, in the other direction, respond best to high frequencies. Obviously, many more features are also represented in this area. In comparison to the core, which again is the primary auditory cortex, the belt and parabelt areas receive diffuse inputs from the thalamus and the core regions, and as such, they process more complex information. This nuance between the core, belt, and parabelt regions underlies an important point about the auditory system that we've been seeing throughout our conversation, which is that stimulus selectivity increases as the pathway progresses. In other words, as auditory information, which was mostly about the frequency and amplitude of sounds, moves from the cochlea to the next brain region to the next, the transfer of information compounds all the previous integration and makes it so that neurons in the secondary auditory cortex can be very specific and process speech to discriminate voice, for example. In addition to this idea that information processing becomes more and more complex as the information moves through the pathway, the secondary auditory cortex is believed to have key regions that initiate pathways to integrate auditory information with the rest of the brain. At the moment, there are two key streams that are believed to originate in belt and parabelt areas, namely the dorsal or aware stream and the ventral or what stream. The dorsal stream is believed to move through the parietal and frontal cortex to aid in sound localization and auditory motor integration, while the ventral stream goes through the memory areas in the temporal lobe and aids in sound identification. A good example of such a stream existing is the relation between Wernicke's area and Broca's area. Indeed, Wernicke's area is located in the temporal lobe near the auditory cortex and is believed to be responsible for speech comprehension. In other words, you can think of Wernicke's area as a very specialized belt region that deals with understanding speech. On the other hand, Broca's area is located in the frontal lobe near the motor cortex and is important for the production of speech. The two areas are known to be connected together via the arcuate fasciculus and thus work together to facilitate a verbal dialogue that is coherent and fluid. Relationships of that kind between an auditory area and an area of another system is obviously not unique. The auditory cortex is known to receive extra influence from vision and the other senses, reward areas, memory, and higher-order cognitive areas like attention and choice. Another important aspect to keep in mind with the auditory cortex is that it provides a massive amount of descending projections back to basically all the subcortical structures we covered. These feedback or top-down connections are believed to improve auditory processing by modulating all the downstream areas. Finally, I want to mention that the auditory cortices, whether primary or secondary, are different between the right and left hemisphere. If we take Wernicke's area, for example, it is known that this area is predominantly lateralized to the dominant hemisphere that performs speech-related functions. Besides speech, some reports seem to point out that music processing might also be lateralized to a given hemisphere. In all, hopefully this illustrates how complicated it all becomes when we reach the cortex and how little we know in the grand scheme of things. Hopefully in a few years, major advances will be made and will provide answers to some mysteries we currently hold. In any case, this concludes our discussion on the auditory system. Throughout this discussion, we focused on the anatomy of the ear, the transduction mechanisms inside the inner ear, and finally, how it all comes together in the central nervous system. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. Thank you again for watching, and I'll see you in our next discussion.